Hello students, in this lecture we are going to discuss about the next production of primary metabolite, that is enzymes. Specifically, now there are different types of enzymes like amylase is there, protease is there, lipase is there, cellulose is there, hydrolase is there, etc, etc. There is wide variety of enzymes which is used for the production of certain different products. Then that products may be chocolate, cheese, yogurts, etc, etc, and this all the production is done by using microorganisms, which includes bacteria, fungi, yeast, mold, etc, etc. So this is a wide variety of the enzymes. Specifically in this lecture we are going to discuss the production of enzyme amylase. Amylase was the first enzyme produced at the industrial level. Before going to the actual production of amylase, let's see the general introduction about the enzymes. Enzymes, these are the these enzymes enhance the rate of the reaction. Enzyme carry out various metabolic processes, various biochemical processes. Enzymes are required in very minute amount, and these are reusable, recyclable. Okay, so this is the general property of the enzymes. Then different microorganisms contribute for the production of the enzymes, like fungi makes the contribution of 60% among the entire microbial world. Bacteria contributes about 24%. Yeast contribute about 4%, streptomyces contribute about 2%, higher animals contribute about 6%, and plants contribute about 4% of the production of the enzymes. Okay, so let's start with the amylase. What are amylase? First of all, amylase, these are the hydrolase enzyme which have been used since many years. And this amylase, these are pancreas and salivary gland make the enzyme amylase. So there are two different types of amylase. Alpha amylase is there and beta amylase is there. The main function of the amylase is it breaks down the starch into monosaccharide. Okay, amylase, what it does, it breaks down the starch into monosaccharide or into simple sugars. Now what is starch? Okay, now starch, it is composed of two types of polymer chain known as amylose and amylopectin. Now question arises, what are these amylose and amylopectin? See, these are the amylose, it is the linear chain of glucose residues. Okay, linear chain of the glucose residues, and this glucose residues are joined together or interlinked together by the alpha 1,4 glycosidic bond or alpha 1,4 glycosidic linkage. Okay, and amylopectin, it is the branch structure linked by both alpha 1,4 glycosidic linkage and alpha 1,6 glycosidic linkage. If we will see the structure of this starch, the starch is made up of two units, amylose and amylopectin. Amylose and amylopectin, they are nothing but the glucose residues attached together by the alpha 1,6 linkage and alpha 1,4 glycosidic linkage. Okay, so this is the स्टार्च एंड दिस अमाइलेज एंजाइम इट ब्रेक्स डाउन द स्टार्च. अमाइलेज क्या करता है? स्टार्च को ब्रेक डाउन करता है. स्टार्च को ब्रेक डाउन करता है मतलब कि क्या करता है? ग्लूकोज रेसिड्यूज की जो चेन है उसे ब्रेक डाउन करता है. मतलब कि क्या करता है? वो बॉन्ड को ब्रेक करता है. ओके, इट ब्रेक द बॉन्ड एंड जो पॉलीसैकेराइड चेन थी ग्लूकोज रेसिड्यूज की उसको वो मोनोसैकेराइड में कन्वर्ट करता है, मतलब इनटू द सिंगल शुगर रेसिड्यू यूनिट. Okay, means what? Starch break down into dextrose, that is the disaccharide sugar. Dextrose, it breaks down into the maltose, and maltose finally break down into the glucose, and this is ये breakdown carry out होता है by the enzyme amylase. Okay, two types of amylase है, alpha amylase and beta amylase. Among these two type of enzyme, alpha amylase is found in the human beings. Okay, The function is the same, that is it break down the starch into glucose. Now this amylase and beta amylase, it is used for making syrups, okay. And this amylase, these are present in the saliva as well as the pancreas. These are somewhat sweetery, so they are used to make artificial sweeteners, okay. Saliva, it contains the amylase. Why it contains the amylase? To break down the carbohydrate into the sugars, okay. So they are used in the industry to make the artificial sweeteners. That is the role of the amylase in the industries. Getting my point? So that is the alpha amylase and beta amylase. If we will see the production of this alpha amylase, then the mainly production which has been involved, see production of alpha amylase sources agar dekhe jaye to, this is, as I told, that this enzyme has been produced by a wide range of the microorganisms including bacteria, fungi, yeast, etc, etc. So the production has been carried out by the 4 steps. That is, first one is selection of microorganism, second is selection of raw material, third is fermentation process, and fourth is downstream processing or recovery, okay. So these are the 4 steps for the production of the amylase. Now let's start with we will see each step in detail. First, I have told you that is selection of microorganisms. Now microorganisms, it may be mostly in the bacteria. Bacillus subtilis is been used. By using Bacillus subtilis, the amylase enzyme is been produced at the temperature range of 40 to 45 degrees Celsius. And if you want the more production, then temperature is been kept in the 20 to 37 degree Celsius, okay. This is in the case of the bacteria. Even we can produce the amylase with the help of the fungi. The fungi which are used are Aspergillus niger and Aspergillus oryzae, okay. So these two fungi are being used for the large scale production of the amylase, okay. Why these two species are being used? Because they have the ability to produce the extracellular enzymes. See, enzymes दो form में produce होते हैं. First है intracellular, second है extracellular. Intracellular में inside the cells enzymes are being produced, और extracellular में outside the cells the enzymes are being produced. तो fungi ये extracellular enzyme produce करते हैं. And the growth is very mat, okay. Mycelium grow होता है, hyphae grow होती है, और inside that our required or desired enzyme is been there. जब हम downstream processing देखेंगे तब हम mycelium से अपना enzyme कैसे purify करते हैं वो भी हम देखेंगे, ठीक है. So that is the main concept. By using these two species we produce the amylase enzyme. Next step is selection of raw material. Now this amylase natural medium में तो grow होता नहीं है, okay. So we provide the synthetic media, and this synthetic media is made by using certain ingredients like corn starch is there, okay. Corn starch होता है. Corn starch, it act as the carbon source. Then KCl is there, that is potassium chloride. It helps to maintain the pH. Then calcium chloride हम provide करते हैं. Magnesium chloride into water हम provide करते हैं. तो these are the certain ingredients which are used for the production of the amylase. The main ingredient is corn starch, okay. So that has been there. Third step is the fermentation process. Now fermentation process has been carried out in 2 ways. First is a solid state fermentation, and next is submerged fermentation, okay. Now submerged fermentation has been carried out with the help of using solid state substrates. तो कौन कौन से substrates use किए जाते हैं? The substrates which are being used are, okay, the substrates which are being used, these are the bran, bagasse and paper pulp. Now what is bran? Bran, it is the, we can say yes, we can say भूसा, और जिसे हम भूसा कहते हैं, okay. गेहूं का भूसा हम कहते हैं, व्हीट का भूसा होता है, दैट इज द ब्रैन. Bagasse means this is the dry dusty pulp that remains after juice is extracted from the sugar cane. Okay, it means it is the dry part after the extraction of the juice from the sugar cane. So that is the bagasse. Paper pulp, yes, paper pulp is been used. So these are the sources which act as the substrate for the production of the amylase. So it is known as the solid state fermentation, means a solid content are being used for the liquid medium, and liquid medium is the submerged fermentation. Okay, this fermentation technique is suitable for the microorganisms like bacteria, उन्हें high moisture, जिन्हें high moisture content की ज़रूरत होती है for the growth. Okay, plus this technique is used for the production of the secondary metabolites like antibiotics are there, हार्मोन्स आर देयर. ओके, सो फॉर द सेकेंडरी मेटाबोलाइट यूज किए जाते हैं. प्लस सबमर्ज्ड फर्मेंटेशन इज ऑल्सो मीन्स इन दिस मेथड if suppose we want to produce the amylase with the help of the fungi. Okay, with the help of the fungi Aspergillus niger, तो हम क्या करेंगे, इनोकुलम प्रिपेयर करेंगे. ओके, इनोकुलम इज प्रिपेयर्ड बाय कल्चरिंग द फंगस. ओके, इनोकुलम प्रिपेयर किया जाता है, फंगस के स्पोर्ट्स लिए जाते हैं इनटू द स्मॉल फ्लास्क कंटेनिंग द लिक्विड मीडियम, और फिर धीरे धीरे उसकी ग्रोथ बढ़ाई जाती है. दैट जो स्मॉल फ्लास्क का कंटेंट है वो लार्जर फ्लास्क में डाला जाता है, सो दैट द प्रोडक्शन विल बी इंक्रीज. ओके, फिर लार्ज फ्लास्क से बड़े जार में डाला जाता है, एंड देन फाइनली वो इनोकुलम डेवलप हो जाता है विदिन 7 to 8 days, एंड देन दैट इनोकुलम इज ट्रांसफर्ड इनटू द फर्मेंटर और बायोरिएक्टर टू कैरी आउट द एक्चुअल प्रोडक्शन ऑफ द अमाइलेस. ओके, सो दिस इज ऑल द सबमर्ज्ड कल्चर मेथड में ये सब किया जाता है. ओके, and once your inoculum is been increased, you add that inoculum into the fermenter. वो fermenter में क्या होता है, your raw material is been there, your ingredients are been there, and then you maintain temperature between 30 to 45 degrees Celsius, and after certain time 72 hours, okay, after 72 hours of the fermentation period, the recovery, that is the last step, downstream processing is been done. Okay, recovery में क्या किया जाता है, आपका प्रोडक्ट सेपरेट आउट किया जाता है. अब आपके फर्मेंटेड प्रोडक्ट में क्या क्या है, आपका एंजाइम है, दैट इज अमाइलेस, मीडिया इज देयर, सिंथेटिक मीडिया, एंड माइसेलियल फंजाई है जहां पर आपका एंजाइम है. ओके, माइसेलियल फंजाई. सो इट इज इंपॉर्टेंट टू फिल्टर दैट. ओके, सो माइसेलियल फंजाई फिल्टर किया जाता है. फिल्ट्रेशन इज बीन डन. बाय यूजिंग फिल्ट्रेशन आवर अमाइलेस इज बीन सेपरेटेड. ओके, फिर amylase separate is been separated, that filtrate contains the amylase, but still it is not in the purified form. Then we add the ammonium hydroxide. Okay, हम क्या ऐड करते हैं, amylase में ammonium hydroxide ऐड करते हैं, which form precipitate with the amylase. precipitation form होता है, that precipitate is been filtrate. ओके, वो फिल्टर किया जाता है बिकॉज इट कंटेन्स द अमाइलेस, एंड देन इट इज क्रिस्टलाइज्ड, उसका क्रिस्टलाइजेशन होता है विच गिव्स द फॉर्मेशन ऑफ द क्रिस्टल्स. ओके, दैट क्रिस्टल्स आर ऑफ द अमाइलेस. तो अमाइलेस हमें फाइनली मिलता है क्रिस्टल्स फॉर्म में. सो लाइक दैट प्यूरिफिकेशन और डाउनस्ट्रीम प्रोसेसिंग इज बीन डन. Now the question arises, how you will determine the enzyme activity. Then there are various methods to determine the enzyme activity. Among this, the one is been mentioned, that is DNS method, which stands for dinitrosalicylic acid method. अब इस मेथड में क्या किया जाता है, DNSA actually this agent is used for the detection of the enzyme activity. सो जो आपका एंजाइम सैंपल है उसके स्टॉक सोल्यूशन, उसका सोल्यूशन बनाया जाता है. मीन्स द स्टॉक सोल्यूशन इज बीन प्रिपेयर्ड, एंड देन दैट स्टॉक सोल्यूशन इज मिक्सड विद दैट एंजाइम सोल्यूशन. इंक्यूबेशन किया जाता है, फिर DNSA रिएजेंट ऐड किया जाता है. ओके, DNSA रिएजेंट क्या करता है, इट स्टॉप्स द एबिलिटी ऑफ बाइंडिंग सबस्ट्रेट टू द रिएक्टेंट. Okay, so the DNS is been added, then incubation होता है. incubation के बाद फिर रीडिंग ली जाती है. रीडिंग एट दी ऑन टू द कैलोरीमीटर एट 540 नैनोमीटर, और फिर एंजाइम एक्टिविटी मेजर की जाती है. ओके, सो टू मेजर द एंजाइम एक्टिविटी इज वेरी डीप पार्ट, इट इज a very detailed part. The most important part to understand at the SY level is the production and the organisms used, the raw materials used. So just keep in mind that enzyme activity is determined by the DNS method. Okay, this is the most important reagent used for the determination of various enzyme activity, not only amylase but also invertase but also cellulase. So ये reagent specifically enzyme activity determine के लिए ही बनाया गया है. Okay, so this is all about the enzyme amylase. What we have discussed general introduction about the enzymes, general introduction about the amylase, the production method of amylase, which consists of the 4 steps. The first one is selection of microorganisms, selection of raw material, fermentation process, which is carried out by using submerged fermentation with the help of the solid state substrates are being used. Okay, so with the help of this solid state substrate, and the last one was recovery or downstream processing. Thank you.